Hello guys, we will talk about cell wall of bacteria. So in the first video, please go back to the first video if you haven't watched yet. It will help you to know about the structure of bacteria, the different types of bacteria, different shapes of bacteria, the nutrition and all that, all the general ideas you will get about the bacteria in lecture 1. So this is the second part where we will start with cell wall of bacteria. Okay. We will start with cell wall of bacteria. So first of all, we need to know that the cell envelope, the cell envelope is made up of plasma membrane and cell wall. There is a cell envelope outside the bacterial cell, and that is made up of plasma membrane and cell wall. And the bacterial cell wall, it differs from other organisms by the presence of peptidoglycan. Bacterial cell wall have a special composition and it is made up of peptidoglycan. What is peptidoglycan? It is made up of peptidoglycan. And this peptidoglycan, it is located outside of cytoplasmic membrane. So if this is a cell, if this is a cell membrane, and outside the cell membrane is the layer of the cell wall. This is cell wall in case of bacterial cell, and this cell wall is made up of, here it's made up of peptidoglycan. Now what is this peptidoglycan made up of? This is made up of, this peptidoglycan is made up of some polysaccharides. So it is having polysaccharide backbone. It is having polysaccharide backbone. It is having polysaccharide backbone, and this backbone, this polysaccharide backbone, it consists of alternating N-acetyl glucosamine and N-acetyl muramic acid. N-acetyl glucosamine, which is said to be as NAG, and N-acetyl muramic acid. This is called as NAM. So this polysaccharide is made up of alternating NAG and NAM units, and they are in equal amounts. So I write here in equal amounts. Both NAG and NAM, they will be present in equal amounts. What are these NAG and NAM? They are the polysaccharide units which make a polysaccharide backbone, and they are alternating. They are alternating. NAG alternates with NAM. So this way, if I draw the structure, it looks somewhat like this: NAM and NAG. With NAM, there is a tetrapeptide attached. Tetrapeptide. And with tetrapeptide, there is another tetrapeptide bonded. Got it? And this is a kind of bridge which is known as peptide interbridge. So a peptidoglycan is in with direct cross-linking. And you can see that. So one thing you need to know that for gram-positive bacteria, there is a direct cross-linking between this tetrapeptide. There is a direct cross-linking between this tetrapeptide. I am not writing it here. there is a direct cross-linking. This cross-linking is direct. Okay, direct cross-linking. But for gram-negative bacteria, this is gram-negative bacteria. This is for gram-negative bacteria. There is direct cross-linking between the tetrapeptides. But for gram-positive bacteria, there is a peptide bridge. Peptide bridge is there which links the two peptide. Means in intermediate there is interpeptide bridge. There are some peptide which are there which links this two tetrapeptide. Clear? So I draw here for both. In case of gram-positive bacteria, how it will look like, and in case of gram-negative bacteria, how it will look like. In case of gram-negative, there is a direct linkage between tetrapeptide. But in case of gram-positive bacteria, there is tetrapeptide, and then there is, in between, there is interpeptide bridge. There are peptide molecules which are there here, but here it's a direct linking. There are no peptides in between. Here no peptides are there in case of gram-negative bacteria, but here peptides are there. Interpeptide means peptides are there between two tetrapeptides. Got it? So it will not be interpeptide, it will be peptide interbridge. Okay. So next comes, what is the role of peptidoglycan? Why is peptidoglycan required? Okay, what do peptidoglycan do for the bacteria? So this peptidoglycans, they are porous in nature. Okay, and they are not considered as a permeability barrier for small substrate. Okay, and there are mainly two types of bacterial cell walls. Bacterial cell wall are of two types. Okay, two types of bacterial cell walls. One for gram-positive bacterial cell wall, and another for gram-negative bacterial cell wall. Okay, and they are different. How can we differentiate between these two? They can be differentiated based on gram staining. How we will know which bacteria is gram positive and which bacteria is gram negative? So they can be differentiated based on gram staining. Okay, and one thing I would like to mention, that the tetrapeptide which was there, which we talked about, this tetrapeptide have alternating D and L amino acid. This tetrapeptide have alternating D and L amino acid. Like for example, with NAM it is attached L alanine, then it is D glutamate, then that is L lysine, then it is D alanine. Okay, so this tetrapeptide, they have alternating L and D amino acids. You can see here. One is L, then D, then L lysine, then D alanine. So this is how they are alternating. Okay, so I am removing this tetrapeptide part here. So moving forward to the next part. So this we need to know about some important characteristics about the gram positive bacteria and gram negative bacteria. Okay, some important characteristics we need to know about gram positive and gram negative bacteria. So first of all, it's about cell wall. First of all, it is about cell wall. As I already discussed, now this is again a kind of assignment. Okay, self-test, which you will do with me right now. Okay, so for gram positive bacteria, how is the cell wall? How is the cell wall? The cell wall is made up of peptidoglycan, and if the peptidoglycan is there, if we stain it with crystal violet, okay, if we will stain it with, here I write gram reaction. Instead of cell wall, I write here gram reaction. Okay, they will retain crystal violet. If I will add crystal violet stain, if I will stain both the mixture of cells, gram positive bacteria will retain crystal violet dye and they will become purple or blue. But in case of gram negative bacteria, they will not take up any stain. Okay, they will not take up any stain, so they are decolorized. Okay, so they are counter stained to check whether they take another stain or not. So counter stained using safranin. So if they take the counter, in case of cell wall, the cell wall is smooth in case of gram positive bacteria. Okay, the cell wall is smooth and it is thick in case of gram positive bacteria, and the cell wall is wavy and thin in case of gram negative bacteria. Okay, and the peptidoglycan layer, it is thick and multi-layered. Thick and multi-layered in case of gram positive, and it is thin and single layered in case of gram negative bacteria. It is single layered because peptidoglycan layer is very thin. So teichoic acid, another chemical compound which is present in case of gram positive bacteria and it is absent in gram negative bacteria. Then another periplasmic space. Periplasmic space. In case of periplasmic space, it is absent in gram positive bacteria but present in gram negative. Then the outer membrane, very important. Outer membrane. This outer membrane is present in case of gram negative bacteria. Okay, then comes porins. Are porins present in gram positive or gram negative? So porins are present in gram negative bacteria. Where? In outer membrane. Next comes lipopolysaccharide layer. Lipopolysaccharide layer, it is none, it is not there, but it is very high in case of gram negative bacteria. Okay, then comes another organelle, mesosome, which is the infolding of cell wall. So mesosome is prominent. It is prominent in case of gram positive bacteria, but it is not prominent in gram negative bacteria. Again, another one, flagellar structure. Flagellar. Here again gram positive and here gram negative. So another comes flagellar structure. So in case of flagellar structure, there are two rings in basal body. There are two rings in basal body. Okay, so these small things we need to remember. In case of gram negative, there are 4 rings in the basal body. Next comes the toxins. Which are the toxins produced by each of these bacteria? Exotoxins are produced by gram positive bacteria and endotoxins are produced by gram negative bacteria. It can be endotoxin or it can be exotoxin. Both can be produced. Next is resistance to physical disruption. Okay, resistance to physical disruption. Which are more resistant to physical disruption? So gram positive bacteria, they are more resistant, they are highly resistant, and here they are less resistant. Okay, next is cell wall disruption by lysozyme. Cell wall disruption by lysozyme. Which one is easily disrupted by lysozyme? Which one you think it can be easily disrupted by lysozyme? Okay, so this can be easily disrupted, high, and they are not easily disrupted by lysozyme. They require a pre-treatment to destabilize the outer membrane, because outer membrane is very much resistant, so they are not so easily disrupted by lysozyme. So they require pre-treatment to destabilize outer membrane. To destabilize outer membrane. Then susceptibility to penicillin or sulfonamide. So susceptibility, just a moment, susceptibility to penicillin and sulfonamide. So which one is more susceptible and which one is less? So they are more susceptible. So remember, whenever outer membrane is there, it makes the cell very resistant. Okay, then susceptibility to another antibiotic, streptomycin, chloramphenicol, and another is tetracycline. So these are the ones with which the gram positive bacteria are less resistant, less susceptible, and these are highly susceptible. So with penicillin and sulfonamide, the gram positive bacteria are susceptible, but with streptomycin, chloramphenicol and tetracycline, the gram negative bacteria are susceptible. Okay, next, inhibition by basic dyes. Inhibition by basic dyes. So with basic dyes, which one is inhibited more easily? So gram positive bacteria are highly inhibited, they are less inhibited. Then susceptibility to anionic detergents. Susceptibility to anionic detergents. So which one is more susceptible to anionic detergents? They are highly susceptible to anionic detergents. Gram negative bacteria are less. Next is resistant to sodium azide. Sodium azide. Which one is more resistant? Gram positive bacteria are highly resistant. Next is resistant to drying. Which one is easily dried? Which one is easily dried? It is gram positive bacteria. They are resistant to dry. They are not easily dried. Okay, sodium azide, gram positive bacteria are resistant to sodium azide. So you have to make a table which one is resistant to which of the chemicals. Okay, so coming to plasma membrane, coming to another important topic. So now we discussed about cell membrane. Now let's start with plasma membrane. So inner to the cell wall is plasma membrane. Okay, inner to the cell wall is plasma membrane. So there was one term named as teichoic acid. So what is teichoic acid? Here I should write what is teichoic acid. So teichoic acid, they are the polymer of glycerol. They are the polymer of glycerol. Okay, joined by phosphate bond. Polymer of glycerol joined by phosphate bond. Okay, so next let's start with plasma membrane. So how the structure of a gram negative bacteria looks like? So outside is the this is if this is cell membrane, okay, head and tail like this, you all know how the structure looks like. Then if this is the lipid layer, if this is the lipid layer, if these are the lipid layer, okay, which are what? These are lipopolysaccharide, LPS, and this is outer membrane, this is outer membrane, and here are some channels named as porin channels. Okay, so next to the outer membrane is what? It is cell wall. Inner to the cell wall is the plasma membrane, which is again double layered. This way is the structure of the plasma membrane. Okay, and this is periplasmic space. So this whole structure is of gram-negative bacteria. Okay, so this is what cell wall, outer membrane, then cell wall, then again this is cell membrane or plasma membrane. Okay, so the plasma membrane, what it is composed of? It is composed of phospholipid, made up of phospholipid bilayer. As you can see here in the last structure, this one, phospholipid bilayer. Okay, so whatever function the cell membrane has, it is as the same membrane as in bacteria, and it's a permeability barrier. Okay, this is a permeability barrier for most of the molecules, and they are where the transport molecules are located. So transport molecules or transporters are located on plasma membrane. Okay, and the fatty acid which are there in the plasma membrane, it can have wide variety of fatty acid within the membrane. So wide variety of fatty acids can be there in the membrane. Wide variety of fatty acids can be there in the membrane, in their membrane. So along with typical saturated and unsaturated fatty acid which are present, bacteria can contain fatty acid with additional, they can have fatty acid with additional methyl group, hydroxyl group, or even cyclic groups, along with saturated and unsaturated fatty acid. Okay, so they have some functional group attached along with saturated and unsaturated fatty acids. So what is the role of these? It maintains the optimum fluidity, for maintaining optimum fluidity. Got it? So as a phospholipid bilayer, the lipid portion which is there, the lipid portion of the outer membrane, this one, the lipid portion of the outer membrane, which is this part, the lipid portion of the outer membrane, which is this part, the LPS of the outer membrane, this LPS is impermeable. This LPS is impermeable to any charged molecules. Okay, impermeable to charged molecules. This LPS is impermeable to charged molecules, and the porins which are present, okay, the porins which are present, which are channels which are present in the outer membrane, what do porin do? It allows for passive transport. It helps in passive transport. Passive means without energy. passive transport of ions, sugars, amino acids across outer membrane. So once these take entry into once the molecules which are transferred to the porine, once they take entry, they remain in the periplasm. They remain in the periplasmic space. Okay, they remain in the periplasmic space, and which is the region between the cytoplasm and the outer membrane. Okay, so periplasmic region, it is a region between periplasmic space, it is a region between cytoplasmic and outer membrane. Now if we go into detail about periplasm, what does it contain? What does periplasm contain? This periplasm contains peptidoglycan. It also contains peptidoglycan and some proteins. What are these proteins responsible for? For substrate binding. They are responsible for, these proteins are responsible for substrate binding or hydrolysis or reception of any extracellular signal. These kind of proteins are found in periplasmic space. And what is the chemical nature of this periplasm? They are gel-like structure. They have a gel-like state, okay, due to the higher concentration of proteins. Why? Due to the higher concentration of proteins. Okay, so number one is they contain peptidoglycan and some protein. They are gel-like state due to the higher concentration of proteins and peptidoglycan. Proteins and peptidoglycans. So since they are located in cytoplasmic, between cytoplasmic and outer membrane, the signals which are received and the substrate which are bound, they are available. They can be transported across the cytoplasmic membrane using some transporting signals embedded there. Okay, so this is about periplasmic space. Now coming to fimbriae and pili. We have done a little about pili. Okay, we have done a little about pili. So fimbriae are what? Fimbriae, they are called kind of attachment pili. What are fimbriae? They are kind of attachment pili, and they are protein tubes. They are kind of protein tubes that extend from outer membrane, which extend from outer membrane. Okay, that extend from outer membrane in many members of proteobacteria. In many members of proteobacteria. And these fimbriae they are short in length and they are present in high numbers about the entire bacterial surface. From all over the bacterial cell surface they come out. And what is the role of the fimbriae? They facilitate the attachment. Okay, they facilitate the attachment of the bacterium to the surface. To any surface, it helps in attachment. Okay, say for example to form biofilm. to form biofilm, or to other surface, or attachment to other surface, or to other surface such as attaching to animal cells during pathogenesis. So these are the function of fimbriae. What other functions are there? There are few organisms, example Mixococcus, they use fimbriae for motility. It is mainly for attachment, okay, but exception is there, okay, exception is there, which is Mixococcus. Which uses fimbriae for motility. Okay, to facilitate the assembly of multicellular structures. Okay, to facilitate the assembly of multicellular structures such as fruiting body. Such as fruiting body. So this is for fimbriae. So what about pili? So they are similar in structure to fimbriae. They are much longer than fimbriae. And they are present in low number. Okay, and they are involved in the process of bacterial conjugation. What is the role? They are involved in the process of conjugation, bacterial conjugation, where they are called conjugation pili or sex pili. Or sex pili. Pili or pili. So these pili, they are absent in gram-positive bacteria. They are absent in gram-positive bacteria and present in gram-negative bacteria. Okay. So this was about pili and fimbriae. The next part we need to know about the bacterial DNA and plasmids. Bacterial DNA and plasmids. Okay, so the bacterial DNA, it is not enclosed inside of a membrane bound nucleus, but instead they are present in cytoplasm. The bacterial DNA is present in bacterial cytoplasm. This bacterial DNA is present in bacterial cytoplasm, and this DNA is not packaged. Okay, this DNA is not packaged using histones or chromatins as in case of eukaryotes, but instead they exist in highly compact structure. But exist in highly compact supercoiled structure. Supercoiled structure. And the precise nature is still not clear. And these bacterial DNA, they are circular in nature, and along with this chromosomal DNA, this bacterial DNA, we will say it as chromosomal DNA. So along with chromosomal DNA, there is small independent. So along with chromosomal DNA, there is a presence of another non-chromosomal DNA. There is a presence of small independent, which is independent autonomous replicating, pieces of DNA called plasmids are present. So these plasmids are not chromosomal, they are extra chromosomal. Okay, they are not necessary for the function, but their presence is advantageous. Okay, so these plasmids, they often encode advantageous traits. They often encode for traits that are advantageous. But they are not essential. But they are not essential for the bacteria. And these plasmids, they can be easily gained or lost. They can be easily gained or lost by the bacterium because they are not required. Okay, they are just having for advantageous character, or they can be gained or lost, or can be transferred from one bacteria to another. Transferred between the bacteria by horizontal mode of transfer. Horizontal mode of transfer. Okay, so horizontal gene transfer. Got it? Through the way of horizontal gene transfer by formation of conjugation tube, the genetic material of the plasmid can be transferred from one bacteria to another. So these plasmids, they are said to be as, so these plasmids are extra chromosomal DNA in the bacterial cell. Okay, so this was about plasmid. Next we come to ribosomes and other multi-protein complexes. And other multi-protein complexes. So in most of the bacteria, the most numerous intracellular structure is ribosome. So most numerous is ribosome, which is the site of protein synthesis. Site of protein synthesis in all living organisms. Okay, and the prokaryotes have 70S ribosome, and this S is Swedberg unit. S is equals to Swedberg's unit, which is according to the name of the scientist. Okay, and while eukaryote have 80S, and this 70S, it is made up of 50S plus 30S. Due to some overlapping structure, it becomes 70S instead of becoming 80S. And this 50S ribosome unit, it contains 23S rRNA plus 5S rRNA. And 30S ribosome, it contains 16S rRNA. And these rRNA molecules, they differ in size in eukaryotes. Okay, and they are complex with a large number of ribosomal proteins, the number and type of which can vary slightly between organisms. Okay, there are large number of ribosomal proteins, and the number of proteins and the type of the proteins, it can vary slightly between the organisms. So this was about ribosomes. Another thing we need to know about bacteria are nutrient storage structures. So what are the nutrient storage structures in bacteria? They are, number one, inclusions. So these inclusions, they are considered to be non-living components of the cell. They are the non-living components of the cell, and they do not possess any metabolic activity. So no metabolic activity are there in inclusions. And non-membrane bound, non-membranous, not surrounded by any membrane. And the common inclusions, the most common inclusions are glycogen, lipid droplets, crystals and pigments. And coming to the second one, which are gas vacuoles. So these gas vacuoles, they are membrane bound. These gas vacuoles, they are membrane bound. Excuse me. They are spindle shaped vesicles. They are spindle. So spindle shaped vesicles, which are found in some planktonic bacteria and cyanobacteria. These gas vacuoles are found in some planktonic bacteria and cyanobacteria, and they provide buoyancy by decreasing the overall density. So buoyancy, why it is needed? To keep the cells in the upper layer of the water column. Okay, that is why buoyancy is needed, to keep the cells in the upper layer of the water column. So buoyancy they provide by decreasing the overall density. So why is buoyancy required? To keep the cells in the upper reaches of the water column and to continue photosynthesis. So when they will be in the upper layer of the water column, they will continue photosynthesis by taking up the sunlight directly. So these cells, they are made up of shell of proteins which have high hydrophobicity towards their inner surface, making them impermeable to water. Okay, so this is all about nutrient storage structures of this bacteria. In the next video, we will start with reproduction in bacteria, which is again a very important part, and then we will start with mycoplasma. So we'll start with different types of reproduction, which is vegetative or sexual and sexual methods. Then we will start with mycoplasma.